and welcome, and thank you so much for presenting.
Okay, thank you, Dan. Can you see the screen?
Yep, looks good.
Okay. Thank you, Dan, very much for giving me this opportunity. And thank you, everyone, for joining me today or tonight here. Okay. My lab mainly works on heme metabolism. Heme is an iron-containing porphyrin that is critical for many biological processes. Heme is a cofactor for hemoglobin, myoglobin, cytochromes, cytochrome P450s, catalases, many peroxidases, and other enzymes such as soluble guanylate cyclases, inducible NO synthase. Heme can also bind and regulate the function of a number of proteins, including DGCR8, which is also called PASHA, mTOR2, REV-ERBs, and transcription factors such as BAC1. And these hemoproteins and heme-regulated proteins are involved in a wide range of cellular processes, which include oxygen transport, electron transport, drug detoxification, antioxidant defense, thyroid hormone synthesis, signal transduction, microRNA processing, and circadian clock control, and gene expression regulation. But from a biochemical point of view, free heme is a hydrophobic molecule, and it can be toxic to cells. Therefore, heme homeostasis in cells must be tightly regulated. Most animal cells can synthesize heme through an 8-step conserved pathway using 3 substrates: glycine, succinyl-CoA, and ferrous iron. Iron is a trace metal that needs to be imported into mitochondria for the synthesis of heme and iron-sulfur clusters. The newly synthesized heme is released in mitochondria, but many hemoproteins are present in other cellular compartments. For example, globins are present in cytoplasm. Heme-regulated transcription factors are in the nuclei. Cytochrome P450s are in ER. Catalase is in peroxisomes. Because free heme is hydrophobic and can be cytotoxic, cells need to rely on trafficking pathways to translocate heme out of mitochondria and deliver it to different organelles. My lab mainly works on iron uptake and heme transport. For the iron part, we are interested in identifying new genes that are responsible for efficient iron uptake in developing red blood cells. Developing red blood cells take up iron through the transferrin cycle. So in this cycle, the iron-loaded transferrin first interact with its receptor, TFRC, on the cell surface. And the whole complex is internalized through clathrin-mediated endocytosis. During the maturation of early endosomes, iron is released from the transferrin, and it is exported out of endosomes by DMT1, and it can be imported into mitochondria by mitoferrin. The rest of the complex, transferrin and the transferrin receptor, are recycled back to the cell surface. Once they reach the cell surface, apotransferrin dissociates from transferrin receptor, and they are ready for the next round of iron uptake cycle. When I was a postdoc in Barry Paul's lab, we identified a trafficking molecule called sorting lectin 3, or SNX3, that can facilitate the trafficking of transferrin complex from early endosomes to maturing and recycling endosomes. And more recently, we found another trafficking molecule called GREP, which is a guanine nucleotide exchange factor that can mediate or facilitate the recycling of transferrin complex from recycling endosomes back to cell surface. Developing red blood cells highly express GREP, and the GREP can recruit and activate the downstream small GGPase called RAB8. The activated RAB8 can then recruit the exocyst complex by interacting with Sec15, which is also called Sec15L1. The exocyst complex then promotes the exocytosis of the vesicles, and along with the vesicles is the recycling of the transferrin complex. So when GRAB is deficient, developing red blood cells showed a reduced uptake of transferrin ion, and this led to reduced synthesis of heme and hemoglobin. In zebrafish embryos, knockdown of the GRAB homolog called RAB3IL1 by morpholino oligos can lead to anemia phenotype. And this phenotype can be rescued by co-injection of the mature form of RAB3IL1 mRNA. So this data suggests that GRAB plays a critical role in efficient ion uptake in developing red blood cells. Today, I'm going to focus my talk on heme transport. One of the main challenges in studying heme trafficking pathways is that it is difficult to separate heme trafficking proteins from the factors that affect heme synthesis, because the heme synthesis pathway is complex and it can be regulated by many factors. My PhD mentor, Iqbal Hamza, has found that the roundworm C. elegans does not make heme, but requires heme for growth and reproduction. C. elegans lacks the whole heme biosynthetic pathway, but it has many hemoproteins, including globins, cytochromes, cytochrome P450s, catalases, soluble guanylate cyclases. Because there is a low endogenous heme production, C. elegans need to acquire heme from the food. Once the heme gets into intestinal cells, it needs to be delivered to hemoproteins present in mitochondria and other organelles. In addition, since none of C. elegans cells can make heme, other tissues such as neurons and muscles need to acquire heme from intestinal cells. Because there is no interference of the endogenous heme production, C. elegans provides a clean genetic background and a unique animal model for studying heme trafficking pathways. In C. elegans, the tissue that is most important for heme homeostasis is the intestine. The intestinal cells need to import heme, utilize heme, store heme, and export heme to other tissues. So in order to identify new genes that are involved in heme homeostasis, we performed the RNA-seq experiment on C. elegans intestinal cells. So in this experiment, we first grew the worms at a low concentration of heme, 2 micromolar, optimal concentration, 20 micromolar, and a high concentration of heme, 400 micromolar, for 1 generation. Then we dissected the adult worms, isolated their intestinal cells, and performed the RNA-seq experiment. In this experiment, we found many previously known heme-responsive genes, including HLG1, HLG2, HLG3, HLG4, and HLG7. And these genes were all upregulated when the worms will grow at a low concentration of heme, 2 micromolar here. And in addition to these genes, we found one previously uncharacterized gene, R186.1, that is also highly upregulated when the worms will grow at a low concentration of heme. And this regulation was further confirmed by real-time PCR. So as you can see, 2 micromolar heme upregulated R186.1 gene expression significantly. So we named this gene as the heme-responsive gene 9, or HLG9. To further confirm this heme-regulated expression, we generated a transgenic worm expressing NLS-GFP under the control of the promoter of HRG9. As you can see in these images, HRG9 is exclusively expressed in the worm intestine, and this expression can only be observed when the worms will grow at a low concentration of heme. In C. elegans, HRG9 has a paralog, which is also previously uncharacterized. It is Y80G38.9, and we call this gene HRG10. Transgenic analysis revealed that HRG10 is also predominantly expressed in the worm intestine. In addition, it is also weakly expressed in another tissue called hypodermis. The expression of HRG10 is not significantly regulated by heme concentrations. To find out whether HRG9 and HRG10 are involved in heme homeostasis, we silenced these genes in a heme sensor strain called IQ6011, which was previously generated in Iqbal's lab. In this is a heme sensor strain. It expresses GFP under the control of HRG1 promoter. Because HRG1 is selectively regulated by heme levels, so the heme intensity, the GFP intensity is inversely correlated with the heme levels. Here we use the long heme importer HRG4 as the positive control. So lockdown of HRG4 leads to increase the GFP expression, indicating heme deficiency in this worm. And similarly, we found that lockdown of HRG9 and lockdown of HRG10 both increased the GFP expression, suggesting that these worms are also heme deficient. Panel B is a quantification of the GFP intensity in the worms. To confirm this result, we deleted HRG9 and HRG10 in the worm using CRISPR-Cas9 and crossed the mutant worms with the IQ6011 heme sensor worms. And as you can see in panel A here, knockout of HRG9 and HRG10, or both, all increased the GFP expression, indicating that these worms are heme deficient. We further performed the real-time PCR and confirmed that the endogenous HRG1 expression is also upregulated in the HRG9, HRG10, or double knockout worms. So these results in the heme sensor strains indicated that loss of HRG9 and HRG10 induces heme deficiency response in the worm. To confirm the heme phenotype, we fed the worms with a toxic heme analog called gallium protoporphyrin 9, or GAPP. GAPP can enter the cells through the heme trafficking pathway. So in the presence of GAPP, the wild-type worms are severely growth retarded due to the toxicity. But the HRG9 and HRG10 knockout worms, as well as the double knockout worms, are less sensitive. Most of them can still grow into adulthood. When we increase the concentration of GAPP in the worm culture conditions, the majority of the wild-type worms die within the 4 days. But most of the HRG9 and HRG10 knockout worms can still survive. So these results are consistent with the observations in the heme sensor strain and suggest that HRG9 and HRG10 knockout worms may be heme deficient. So next, we measured the heme content and heme uptake in the worms. So panel A is the total heme content in the worms. There is a low significant difference between the wild-type worm and the HRG9 and HRG10 mutant worms. So here, HRG4 is used as a positive control. And we further measured the heme uptake using N15 labeled heme. And again, we do not see significant difference between the wild-type worm and the HRG9 and HRG10 mutant worms. So these results indicate that the HRG9 and HRG10 mutant worms have normal heme uptake and normal heme content, and further suggests that the heme deficiency phenotype we observed in the gallium protoporphyrin 9 experiments and the heme sensor experiment is not due to reduced heme uptake. So next, we analyzed the intracellular heme distribution using a fluorescent heme analog called zinc metporphyrin, or ZNMP. Compared with the wild-type worm, the HLG9 and HLG10 mutant worms showed increased staining of ZNMP in the intestinal cells. And panel B is a quantification of the ZNMP signal. It has been shown by Iqbal's lab that in C. elegans intestines, ZNMP mainly localizes to lysosome-related organelles, or LROs, which are the heme storage site in C. elegans intestinal cells. So to find out whether zinc metporphyrin also localizes to LROs in HLG9 and HLG10 deficient worms, we used the LRO marker, PGP2-GFP. As you can see in the merged images, ZNMP and PGP2-GFP co-localizes well in both the control worms and the HLG9 and HLG10 RNI worms. To further verify this observation, we blocked the biogenesis of LROs by silencing PGP2 and stained the worms with ZNMP. And yet, as you can see in these images, lockdown of PGP2 leads to reduced staining of ZNMP in both the wild-type worm and the HLG9 and HLG10 double knockout worms. So these results indicate that when HLG9 and HLG10 are deficient, heme accumulates in the storage site, LROs, and less heme is available to the worm. So the worms show heme deficiency phenotypes. To find out whether this heme accumulation is due to increased heme loading or reduced heme mobilization from LROs, we performed pulse chase experiments using ZNMP, or zinc metporphyrin. As a panel A is the pulse experiment, and we found a low significant difference between the wild-type worm and the HLG9 and HLG10 double knockout worms. But as you can see in panel B, the HLG9 and HLG10 knockout worms showed a delayed clearance of ZNMP during the chase. This result suggested that HLG9 and HLG10 regulate heme export from the heme storage site. And this function is similar to that of HLG1, which is a long LRO heme exporter. So to find out whether HLG9 and HLG10 works in the same pathway as HLG1, we silenced the HLG1 in the double knockout worms. As you can see in panel B, silencing of HLG1 further increased the intensity of zinc metporphyrin in the double knockout worms, suggesting that they may function individually or independently. HLG9 has a homolog in almost all eukaryotes, ranging from yeast to mammals. And this homolog has been previously named as Tango2. Tango2 was first identified as one of the genes that regulate protein transport and Golgi organization in a genome-wide RNA screen in fly S2 cells. Sorry, there's one. Okay. So HLG9, HLG10, and Tango2 are all predicted to be soluble proteins without transmembrane regions. Since in C. elegans, HLG9 and HLG10 regulate heme homeostasis, so we asked whether these Tango2 proteins also regulate heme homeostasis. So immunofluorescence assays showed that human Tango2 proteins mainly localizes to cytoplasm and mitochondria. Mitochondria is the heme synthesis site. To find out whether Tango2 regulates mitochondrial heme homeostasis, we deleted Tango2 in HEC293 cells and measured the heme content. We found that knockout of Tango2 leads to increased mitochondrial heme levels, but not in lung mitochondrial fractions. We observed similar results in mouse erythroid leukemia cell or male cells. Here we generated Tango2 knockout male cells and fractionated mitochondrial and lung mitochondrial fractions. And again, we see that knockout of Tango2 leads to increased heme levels in the mitochondrial fractions, but reduced heme levels in the lung mitochondrial fractions. Our collaborator, Amita Reddy, also analyzed the heme levels in a Tango2 knockout yeast strain using a fluorescent heme sensor. This sensor contains 3 modules: the internal standard MK2, a heme reporter EGFP, and a heme binding module. When heme binds to this sensor, EGFP fluorescence is quenched, so it's reduced, and MK2 fluorescence is unaffected. In the mitochondria of Tango2 knockout yeast, the EGFP to MK ratio of the sensor is severely reduced, indicating increased heme in the mitochondria of the Tango2 knockout yeast. And this increase can be largely suppressed by succinyl acetone, which is a heme synthesis inhibitor. In the cytosol of Tango2 knockout yeast, the EGFP to MK ratio of the sensor is significantly increased, indicating heme deficiency in the cytosol. And here, heme1 delta is used as a positive control. So the results in the yeast are consistent with the results in mammalian cells and suggest that Tango2 may be involved in heme transfer from mitochondria. So to summarize this part, we have identified a new heme responsive gene, HRG9. In C. elegans, HRG9 and its paralog HRG10 regulate heme export from the heme storage site, LROs. In mammalian cells and yeast, the HRG9 homolog Tango2 regulates heme export from the heme synthesis site, mitochondria. Since HRG9 and Tango2 are both involved in heme homeostasis, we next asked whether they have the same function. To address this question, we expressed Tango2s in C. elegans. Here, as you can see, knockout of HRG9 and HRG10 leads to increased ZNMP fluorescence. And this phenotype can be significantly rescued by the expression of human Tango2 or yeast Tango2 or zebrafish Tango2 in the mutant worms. This result suggests that Tango2s are functional homologs of HRG9 and HRG10. Because mitochondria and LROs are both enriched in heme, so our results further suggest that HRG9 and Tango2 may transfer heme from heme-enriched organelles. To find out whether these proteins can transfer heme, we performed in vitro heme transfer assays. Here we used isolated mitochondria as a heme donor and apomyoglobin as a heme acceptor. After incubating them, we assayed the heme activity associated with the apomyoglobin, with myoglobin by in-gel heme staining. As you can see in the middle line here, when we incubate the mitochondria and apomyoglobin, there is a low spontaneous heme transfer. Addition of Tango2 to the reaction leads to robust activity of heme associated with myoglobin, suggesting that Tango2 may facilitate heme transfer from mitochondria to myoglobin. We observed similar results with the yeast tangle 2 protein and the zebrafish tangle 2 protein. And the previous slide was done with human tangle 2 protein. And here panel A is yeast tangle 2 protein. And panel B, we added a zebrafish tangle 2. And we observed similar results. Addition of either yeast tangle 2 protein or zebrafish tangle 2 protein, both increased the heme activity associated with myoglobin, indicating that tangle 2s can facilitate heme transfer to hemoproteins. There are two possibilities here for the function of tangle 2 proteins. The first is that tangle 2 may transfer heme out of mitochondria. And the second is tangle 2 may facilitate heme incorporation into hemoproteins. Because tangle 2 proteins do not have transmembrane regions, so it cannot translocate heme across membranes. So we tested whether tangle 2 protein can facilitate heme incorporation into hemoproteins. In this heme transfer assays, we used the free heme instead of mitochondria as the heme donor. In panel A, we used the 10 micromolar heme, and in panel B, we used the 1 micromolar heme. We found that heme can spontaneously bind to myoglobin, and this binding is not further increased by addition of tangle 2. And we observed similar results with 10 micromolar heme and 1 micromolar heme. So this result suggests that tangle 2 may not facilitate heme incorporation into apomyoglobin. So there's only one possibility left. So tangle 2 can help to translocate heme out of mitochondria, but it does not contain transmembrane region. Given that heme is a hydrophobic molecule that can associate with membrane lipids, so next we tested whether tangle 2 can transfer heme from membranes. So in this heme transfer assays, we used the isolated mitochondrial membranes as the heme donor and the apomyoglobin as the heme acceptor. And we found that addition of tangle 2 indeed can facilitate the transfer of heme from mitochondrial membranes to myoglobin. So this result suggests that tangle 2 may function to transfer heme from membranes to downstream hemoproteins. During the heme transfer assays, we noticed that tangle 2 may associate with heme. Here, when we incubate 10 micromolar heme with tangle 2 protein, we found that there is a heme activity associated with tangle 2 protein. To confirm this result, we expressed flag-tagged human tangle 2, flag-tagged mouse tangle 2 in mammalian cells, purified the proteins, and incubated them with heme. We found that heme can associate with both human tangle 2 protein and mouse tangle 2 protein. We further expressed the yeast tangle 2 protein using bacterial expression system and performed the heme binding experiments. We found that yeast tangle 2 can interact with both ferric heme and ferrous heme. And here, myoglobin and lysozyme were used as controls. When heme binds to proteins, there is a shift of the Sorry band. And based on this shift, we were able to titrate the binding affinity by using different concentrations of heme for the binding assays. The results show that yeast tangle 2 protein may bind the ferrous heme with the dissociation constant of around 15 micromolar, and it binds ferric heme with dissociation constant of around 19 micromolar. These results indicate that yeast tangle 2 protein is a low affinity heme binding protein. And taken together, our results indicate that HLG9 and TANGO2 are a family of intracellular heme chaperones. In the heme auxotroph C. elegans, HLG9 and its parallel HLG10 can mediate the heme transfer from the heme-enriched organelle or the heme storage site, LROs. In heme-synthesizing organisms, such as yeasts and vertebrates, the HLG9 homolog TANGO2 can transfer heme from the heme synthesis site, mitochondria. HLG9, HLG10, and TANGO2 may transfer heme from the heme-enriched membranes and transfer heme to downstream hemoproteins or cellular compartments. Besides eukaryotes, some bacteria also have putative homologs of TANGO2. The homolog of TANGO2 has been characterized in a gram-negative bacteria called Shigella oleidensis, and it was also shown to be a heme-binding protein and has been named as a heme trafficking protein A, or HTPA. HTPA can bind heme with a dissociation constant of around 1.2 micromolar. And in the bacteria, it can facilitate heme transfer to the cytochrome C through the cytochrome C maturation system. It may transfer heme directly to one component of cytochrome C, which is cytochrome one component of CCM system, which is CCMB. Many studies have reported that mutations in human TANGO2 may cause an inherited disease. Patients with TANGO2 mutations display pleiotropic symptoms, including cardiac arrhythmias, encephalopathy, rhabdomyolysis, and metabolic crisis. To provide a vertebrate animal model for TANGO2 diseases, we deleted the TANGO2 in zebrafish. Here we use the two gRNAs, and we got a mutant with two deletions. The first deletion is within exon 2, and the second deletion is in the second exon and intron boundary. We found that loss of TANGO2 is lethal to zebrafish, and this phenotype has a maternal effect. The homozygous mutants from the homozygous parents all die during early development. But the homozygous mutants from the heterozygous parents can still survive to later stage. Some of them can even survive to adulthood. We also performed a preliminary analysis on several tissues of the fish, in the fish. And we found that TANGO2 knockout zebrafish displayed signs of brain atrophy, disorganized muscle fibers, and cardiac arrhythmias. And these symptoms are partially similar to the symptoms of human TANGO2 patients. We also did a preliminary analysis on the effects of TANGO2 deficiency on the mitochondrial metabolism. It has been previously reported that mitochondrial heme overload may impair the coupling of mitochondrial respiration. So we analyzed the mitochondrial OCR and the mitochondrial membrane potential using a dye-TMRE. We found that knockout of TANGO2 leads to increased oxygen consumption rate in HEC293 cells. But the mitochondrial membrane potential is significantly decreased in the knockout cells. So taking together, these results suggest that loss of TANGO2 may lead to uncoupling of the mitochondrial respiration. Two recent studies also reported that TANGO2 deficiency may affect lipid profiles. One study was performed in HepG2 cells. So the authors show that knockdown of TANGO2 in HepG2 leads to increase the level of the lipid called lysophosphatidic acid and decrease the level of phosphatidic acid. The other study was done in zebrafish, and the Tango2 knockout zebrafish showed reduced levels of several types of major lipids, including phosphatidylcholine, phosphatidylethanolamine, diacylglycerol, and triacylglycerol. So we also performed lipidomics analysis in HEC293 cells. However, we did not observe significant differences in the major lipids. And this include phosphatidylcholine diacylglycerol, phosphatidic acid, phosphatidylethanolamine, as well as triacylglycerol. So we are still trying to do more experiments to figure out the connection among heme metabolism, Tango2, and lipid metabolism. So finally, I want to thank everyone who contributed to this work, especially our wonderful collaborators. For the HLG9 story, Fengxiu, Zhejiang, and Shuaiqi did most of the experiments for the worm, mammalian cells, and zebrafish. For the GRAB experiments, Mengying, Yuhan, and Kailun did most of the work. Mathilda from Amidoradis Lab did all the functional assays in the East. Iqbal and Xiaojing contributed to the worm experiments. Jiahai, Qingyi, and Weishi helped with the primary feed liver experiments for the red cell development projects. I also want to thank our university, Zhejiang University, and the funding agencies for their support. And I also want to take this opportunity to thank my great mentors, Ph.D. mentor Iqbal Hamza, and my postdoc mentor Barry Paul. And thank you, everyone.